Okay, we're starting. Right, hello everyone. Welcome to the 14th session for BGA24. This session is on the Genome-Note publications pipeline written by Toll IT, namely Beth Yates and Tyler Chafin. Please take it away.
Hi. My name's Beth Yates. I'll be presenting today. I'll just start sharing my screen. And everyone see a slideshow. Perfect. So I'm a bioinformatician in the informatics infrastructure team, and I work with Tyler, who's another bioinformatician who works primarily on our Nextflow pipelines that get used in production. Michu Mafato is also on this call. He's our team leader. So if there's any questions that I can't answer or Tyler can't answer, I'm sure he'll be able to chip in at the end. So I'm going to be talking today about the work that I've been doing primarily for the last 2 years in the Tree of Life project on automating our Genome-Note publications. So I'll just give you an idea of what to expect from this session. The first thing I'll be doing is giving you an introduction to the Tree of Life project and a kind of an overview of where we fit in and what we're doing and what a Genome-Note is. And then I'll give you some more information about how we're actually trying to automate the production of genome notes. And then I'll introduce the Genome-Note pipeline and explain what it does, what inputs it takes, and what outputs it gives. And there will also be an interactive part to this session where you can run the pipeline yourselves on a test genome data set on the GitHub demo. I'll actually probably start that partway through the explanation of what the pipeline is because it will take approximately 10 to 15 minutes to run. So we can be talking through the pipeline while you're running the script yourself. And then I'll give you a little bit more information about other pipelines that we use to generate Genome-Note content and kind of give you an idea of how these pipelines could be linked together to go from a raw genome assembly submission files to a publication that can be sent to a journal describing that assembly. And then I'll give you a little bit, I'll tell you a little bit about the Universal Genome Notes platform, which is a web-based platform that we're building to allow anyone to generate their own genome notes. And it would make use of the pipeline that we're talking about here, but it would be a more kind of user-friendly way of interfacing with it. So first of all, the Tree of Life at the Wellcome Sanger Institute, I guess a lot of you will know about that already if you found your way to BGA in the first place. But essentially what we're doing is we're aiming to better the human condition through large-scale genome sequencing across a lot of diversity. So we are innovating in the generation of high-quality genome assemblies. We're delivering sequence assemblies across the diversity of the Tree of Life. And we're contributing to global and local projects that aim to build a resource for whole biota genomics. And we're developing unique science based on these genome sequences in the areas of evolution, ecology, and genomics. We are part of several large major projects, including the Darwin Tree of Life, which is aiming to sequence 70,000 species from Britain and Ireland. The Aquatic Symbiosis Genomics Program, the Vertebrate Genomics Project, ERGA, the European Reference Genome Atlas, and the Earth Biogenome Project, which is set itself the not significant task of sequencing all life for the future, sequencing all life for the future of life. So to give you an idea of what we do within the Tree of Life, this is what we describe as the genome engine. So this is the entire range of what we cover. So this is everything from sample acquisition and metadata recording, which we do in conjunction with COPO, to DNA barcoding, to extraction of DNA and RNA, and then to genome and transcriptome sequencing, to submission to databases, mainly ENA for the assemblies and the reads, and then from there it gets distributed to NCBI and other groups. We then do curation and scaffolding and some QC checks. Our partners in Ensemble do gene finding and annotation for us. And everything kind of goes onto an open data portal. And the final step in producing an assembly, which is what we're kind of going to focus on here, is the publication of the Genome-Note. So what is a Genome-Note? Essentially, it's a brief scientific article that describes a genome assembly. It acts as an announcement of the genome's availability, and it provides a formal assessment of the assembly quality. It's also a source of contextual information about the species and a way of acknowledging everyone involved in the genome sequencing process. And there's an example over here on the right of a Genome-Note. All of our Genome-Notes from the Tree of Life project are published in Welcome Open Research. They're freely available for everyone to go and see. And they get peer reviewed there as well. So how we're currently producing a Genome-Note is it's a very manual process. We have a Genome-Note editor within the Tree of Life, Karen, who works very hard on producing these documents. She is supported by the infrastructure team and some of the other teams within the Tree of Life that provide data for her. But it's still very much a case of manually editing a Word document to provide the Genome-Note. So what she currently has to do is she has to go to lots of different data sources, things like ENA, internal JIRA databases, to kind of find some metadata about the genome. She also has to collect the collectors of the samples that we used in the sequencing to ask them to provide some introductory text. And then she also has to put in a request for the running of the pipelines for the Genome-Notes and for the blog toolkit plots that were also included within the Genome-Note. And then all of this information gets combined into a Word document. And I think perhaps one of the more kind of problematic things that she has to do is identify potential reviewers for each Genome-Note. And due to the rules that the journals have, this can take quite a lot of time. So that's something that we're also working on, but that's not something we're going to talk about today. And then she submits the completed note document to Welcome Open Research. And that in itself takes some time because it's quite an involved submission form that she has to do. I think we're now approaching about 1,000 Genome-Notes that have been submitted to Welcome Open Research. So all of the time adds up. So why do we want to automate the production of the Genome-Notes? Well, scalability. We're expecting to produce more than 70,000 notes within the Tree of Life project alone. We want to reduce the time it takes to get a note published and minimize the amount of content that the authors and the editors have to provide. We also want to reduce the cost of publication. So we are working with Welcome Open Research, which is an F1000 journal, to develop a way of bypassing traditional journal submission methods and have the submission happen automatically via an API where we will send an XML file of the Genome-Note and some associated image files directly to their server, and from there it will get processed and turned into a Genome-Note on their system. And we also want to reduce the need for in-depth peer review. So as part of the Genome-Note, we calculate a set of assembly quality metrics based on some metrics that were defined in the Vertebrate Genome Project 2021 paper. And we believe that these metrics can kind of act as an automated peer review. So if the assembly meets all of the standards that have been set out, we can reduce the number of manual reviews that are needed for each Genome-Note. And if you want to read more about that, there's a link to the BGP paper here. I can share these slides after this, I'll add them to the Git pod after the session. So currently, one of the first things I did when I joined the project was to kind of go through a Genome-Note and identify the kind of data that it had. And that was kind of classified into three types of data. So firstly, there's data that can be retrieved from the public databases and resources. So this is things like fetching metadata, taxonomic data, annotations from sources like NCBI, ENA, Ensemble, GOAT, which is Genomes on a Tree, which is another resource that's provided by Tree of Life. And then there's data that can be calculated. So we can have our Genome-Notes analysis pipeline run some software and generate some information that then goes into the Genome-Notes. That is things like the assembly quality metrics I mentioned earlier, BUSCO scores, the Hi-C contact map based on the Hi-C data. And then there's data that is more difficult to get automatically. So this is things that vary more between each individual note. So this is things like the background text, which describes a little bit more about the biology of the species. And we tend to rely on experts to provide that. That's not something that we standardize, although there is a standard paragraph that we can use if we can't get someone to give us that background information. And then things like the author list, the references, the species images, things that will be different in each individual Genome-Note. So that's the kind of thing that we're going to rely on our genome editor being able to provide. So I'm just going to go through kind of the anatomy of a Genome-Note and what we have, what this information is, what kind of information it is, and where we can get it from. So there's kind of color coding used here. So any information that we believe needs to be supplied by the person writing the Genome-Note is indicated in orange. Any data that we can kind of fetch from different sources, either things like ENA, NCBI, is in blue, and anything in grey is information that we can have our pipeline generate for us by running software. So I think you can see here, the Genome-Note starts with a title, which basically just gives you the common name and genus and species of the assembly. Then we have the collectors, what we call the GAL, so that's the Genome Acquisition Lab. So they are a set of groups that we work with, and they provide the genome samples to us. They go out and collect the samples and send them in to us, and we do the sequencing. But we need to acknowledge their contributions as part of the Genome-Note. And then the standard information in the abstract and keywords. So that's the information that we can gather from different sources and add to the Genome-Note. Then there's the background, which tends to be more information that's relevant to that individual species. So it'll be why it's been sequenced, why it's important, any references that are relevant, and just general species information. And then we have the specimen images, which are supplied by the collector. And then we describe how we did the sequencing. So we'll give a description of how the specimen was collected, the sequencing that was performed, and any curation that took place once the sequencing was complete. We'll give a description of the final assembly length, the chromosomes, and BUSCO completeness scores. And if the genome has been annotated, we'll give a description of that process based on information that's supplied to us from Ensemble. Now, we don't always have annotation for a genome at the time we publish a Genome-Note. So this is not something that's always included, but if the annotation has happened already, that will go into the Genome-Note. And then we have a table which has most of the interesting information, the metrics in it for an assembly. So this is where you can find things like the bioproject accessions, the sample accessions, the assembly accessions, so that you can then go and look for all of the data in public sources. And that is something else that we rely on for a Genome-Note. Any species that goes through the Genome-Note pipeline must have been deposited in a public database so that we can use those resources to get some of the metadata information that we need. We also list the chromosomes if the species has been assembled to chromosomes, and give things like the size and the GC length. Sex chromosomes are included if they've been identified, and the mitochondrial genome is also included. We have some more detailed information about the methods used for specimen collection, sample prep, and DNA and RNA extraction. We give some information about the sequencing and the assembly pipeline software and processes that we used. And at the moment, a lot of this information is provided by the Genome-Note editor, because it can be quite varied depending on the type of sample that's being processed and the genus that it belongs to. So we might use different extraction methods for plants as opposed to for a mammal. And then we give details about the curation of the assembly. So we have a team of curators that assess all of our assemblies, and they will do some analyses and try and kind of make the assembly the best it can possibly be. And all the changes that they make are tracked in an internal database, and we can pull that information into a Genome-Note. We also think it's important to report all of the software tools and the versions that we used within both the assembly and the analyses afterwards that get used for the assembly metrics, so that everything we do is reproducible. So we report all of the software tools that are used. As part of the Genome-Note, we generate some images. The one on the left here is a Hi-C map. We generate those as part of the Genome-Notes pipeline, and then we post the files that are generated to a public HiGlass server. So there is an interactive version of the Hi-C map that anyone can kind of go and interact with. We also have a separate pipeline that we run for each assembly, which generates blob toolkit plots. And these plots are also included within a Genome-Note, and they're hosted on a public blob toolkit viewer. But that's a separate pipeline to the Genome-Notes pipeline. So we're not going to be running that today. I'll explain how the pipelines all kind of link together later on in this session. We have a data availability statement. All of the data that we produce is publicly available and anyone can use it. And we try and give a complete list of authors and collectives, and within the collectives, you can go and find out who was involved in the sequencing, so that everyone involved is fully credited. Okay, so now we kind of know what a Genome-Note is, and describe how we're using pipelines to generate content for the Genome-Note. So there's two types of data, as we said before, that can come from the analysis pipelines. There's data that can be fetched from the public databases and resources. So this is things like genome metadata. And we have a sub-workflow within the Genome-Note pipeline that goes away and queries a lot of these different sources and pulls information back. So this is things like ENA, GOAT, NCBI, GBIF, where we get some of the taxonomy data, and COPO, who do a lot of the sample brokering for us. So they have a lot of information about how the sample was collected, how it was preserved, and we can kind of pull that information from them and include it within the Genome-Note. And then the other type of data is information that we can calculate. So this is things that will give us an idea, a way of assessing the genome quality. So this is things like the high contact maps, the blog toolkit plots, the BUSCO scores, and the genome statistics. And here I'm talking about things like QV scores and KMA completeness. So within the Tree of Life, we use Nextflow and NF-Core for our production pipelines. And these were selected for lots of reasons, but I think one of the main things is that they're portable, they are cloud compatible. All software dependencies for the pipeline get packaged into public Docker containers, and all parameters in config file are in config files, so there's no hard-coded paths for dependencies, which makes them very reusable and very shareable. So the idea is that any pipeline that we have developed within the Sanger Tree of Life, anyone should be able to take that pipeline and go and run it on their own data, on their own infrastructure, and they shouldn't need to make any changes. So everything, all of our pipelines are freely available for anyone to use. They are shared with the community. We don't want people to be reinventing the wheel and writing their own pipeline that does the same thing. So yeah, we make everything that we're doing available for anyone to use. So as I said, pipelines are available. So we use Nextflow and then of course standards. You can read more about those on the Nextflow website. We host our own pipelines website, which is essentially a mirror of the NF-Core site. And all of the pipelines, including the Genome-Note pipeline, are hosted there. They are also all on GitHub. So if you spot an issue with one of our pipelines, you can open up an issue on GitHub and we can try and get it fixed. Or if you see a way of that you might be able to improve a pipeline, you can open an issue there, talk to us, put in a pull request, and we can collaborate on these pipelines. And all of our pipelines are citable through Zenodo DOIs. So yeah, all the links are here, and I'll share the slides after the session. So this is just what the Genome-Notes pipeline looks like on our website. So for each pipeline, we have an introduction to the pipeline. We describe the pipeline results, the usage docs for the pipeline, any parameters, including those that are required, and any kind of optional parameters, they will all be explained here. And the output docs describes what you should be getting from the pipeline, so the kind of documents that you'll get, the form that they will take. And then there's just some kind of information on the release and the statistics of the pipeline. Okay. So for the Genome-Notes pipeline itself, there's kind of three main input types for the pipeline. So firstly, there's metadata inputs. So this is things like the assembly and biosample accessions, which we then use to go away and query the different data sources to fetch information about the assembly and the biosamples. There's a sample sheet input, which is where you would give a list of all the samples that we want to analyze for the assembly, and some kind of basic configuration inputs, which are options to control how the pipeline analyses run. For example, there may be some steps within the pipeline that we don't want to run, or there may be a step that we need to give a bit more memory for a certain genome. But all of the inputs and their usage are described on the pipeline website. So for the Genome-Note pipeline itself, the required parameters are the first one is input, which is essentially a sample sheet which lists all of the reads for the assembly that we want to analyze. The FASTA file, which is the assembly FASTA file. It can be either zipped or unzipped, and the pipeline can deal with both. The assembly accession, we expect this to be a INSDC GCA format assembly accession, and the biosample accession for the sample that contains the whole genome sequencing. So they are absolutely required for the pipeline to run. There are some other pipelines, there are some other parameters that are expected, but these have defaults that are defined. So you don't necessarily have to override these. So this is things that kind of, again, control more about how the pipeline runs. So this is bin size, which is used to control cooler cloud, which is cooler cloud. I think that should be an A, not an O, but that's part of the Hi-C mapping process. The KMA size, which just defines the size to use for the KMA library. Like I say, there is a default set for that, so you don't have to override that, but the option is there for you to do. And the minus outdir, which should be a path to where you want the results for the pipeline to be saved. So the default for this is results, but that can be overridden. You can tell it to write the results to wherever you want, but you have to give it the full path. There's also things like optional inputs, which you'll see when we run the pipeline. So for example, we don't always use the same biosample to produce Hi-C data as we use for the whole genome data. So there is the option to provide a different sample accession for Hi-C and a different sample accession for the RNA-seq data. But again, these are not necessary. And kind of what's making the Genome-Note automation work is the pipeline outputs a Genome-Note document at the end of it. And that's based on a template that's given to the pipeline to begin with. So when you want to generate your own Genome-Note, you have to work out what you want your Genome-Note to look like. And then you create a template, which is essentially a document which has most of the note content, but with placeholders for the bits of text that vary from assembly to assembly. So things like the assembly name would be a placeholder. The assembly accessions, the biosample accessions, they would all be placeholders within the template. There is a default template, which is the template that we use for genome notes within the Tree of Life, that's within the assets directory of the pipeline. So if you don't provide your own template, it will default to using the standard template that we have, and the Genome-Note will come out looking very similar to what we produce within the Tree of Life. But if there's different sections that you wanted to include within your own Genome-Note, you can override that note template and provide it with a template of your own kind of design. And then there's kind of just other optional inputs. So I'm not given the full list of inputs here, but they are all on the website. So you can provide the pipeline with an email address. So once the pipeline is completed, it will send you an email. It will give you a summary of what happened. And then there's just other kind of, so for Hi-C, if you want to specify the sequence names in the order that you want them to appear on a Hi-C map, you can give a file that will define those, but the pipeline will work without that. So that's an optional input. You can either supply the parameters on the command line, but as you can see, there's quite a few parameters, and that can get a bit unwieldy, a bit difficult to type out. So you can also supply any parameters when you want to run the pipeline in a parameters file. This is either in JSON or YAML format, and you can use the ---- params file flag to kind of define this. So in the demo that we're going to run shortly, there's a JSON file that contains the values that we will be using to run this little test for you. Okay, so the next step is moving on to the live demo. So this is the command that you're going to be using. It's all in GitHub. So I'm going to stop screen sharing the presentation for a moment and open up Gitpod. And if you'd all like to do the same thing to go to your Gitpod page and start launching that. If any of you have any issues with the Gitpod getting up and running, Tyler, I think, is monitoring the Discord and should hopefully be able to answer any questions that you have. Everyone able to open the Gitpod and see the instructions? See something that looks like what I've got in front of me? So within the Gitpod, we've already got Nextflow installed. We have the Genome-Notes pipeline installed. Within the browser, you can see the command that we're going to use to run the session, to run Nextflow. So once it's fully loaded, on the left-hand side, you should see assets, data, docs, Genome-Note, and then some kind of other files that we don't need to take too much interest in. So Genome-Note is actually where we have the pipeline defined. Data, we're not actually going to be using data, but that is the files that it's the same data that we're using in the pipeline run, but we're using the copy that we have on our public S3 bucket. So within assets, you can see the parameters file that we're using today. It's basically telling you where the sample sheet is, where the FASTA sequence is. We're giving it a path to our S3 bucket, but you could have the file locally. So like here in data, we could just give a path to that. We set the coma size to 11. That's just for the purpose of running this pipeline in a speed that's kind of manageable. So the actual genome assembly data here is not the full genome. It's only a couple of chromosomes. The reads that are within the sample sheet. So if I show you the sample sheet, doesn't open that way. There we go. If you open it with the text editor, you can see we have two PacBio files, and they are both subsampled BAM files. They're not the complete data set. This is just so that we can get the pipeline to run within 10 to 15 minutes rather than the several hours that we would normally take for a full assembly. And the same with the Hi-C file, that's a CRAM file. So the Hi-C reads have been aligned to the genome using our read mapping pipeline. But the PacBio whole, so these are the whole genome sequencing reads, they're unaligned. So that's kind of the data inputs. And then to run the pipeline, all you need to do is find the command within the browser, and you can copy that command. And if you paste it in the terminal at the bottom, oh, not what I meant to paste. Oh, that's not working as a direct paste. Okay. Yes, type that in. So I'm going to open that. An external website. copy it from here and then paste. And then if I hit return, you can see that Nextflow will start running. So if you want to give that a go and see if it works for you. While that's running, I'll give you a little bit more information about the kind of what's happening within the pipeline and what kind of outputs to expect. But before I do that, let's open up. So I mentioned that there's a default template within the Genome Notes pipeline. So I'll open up that template for you. So Genome Notes assets is where you'll find the template. And it is Genome Note template.docx. So this is what I mean by a template file. Any kind of placeholders within. So this is what the document looks like before we run the pipeline and fill in the information. So we define placeholders within the template using these double braces. This is just a way of templating. This is based on something called Ginger, which is a templating framework. So we can then provide the template with a set of a file that contains these kind of names and then a value, and then we can combine the template with that list of key value pairs, and the templating system will fill in the placeholders with the value in the file. So at the moment, the Genome-Note pipeline can't produce a completed Genome-Note. So what we're doing with this, we have indicated anything that we expect the pipeline to be able to fill in is in green. And once the Genome-Note has been produced, that information will still be in the document in green, but it will be the value instead of the bracketed placeholder text. But we keep it in green so that you can see what's been changed and what's been filled in. If the pipeline hasn't been able to fetch one of the placeholders, for example, not every species has a common name, the placeholder will remain in the template, and then it's up to the editor to remove that or replace it if they can find the relevant information elsewhere. So yeah, there's also things in blue which we expect the editor to provide. These are things that we can't work out manually. So there's things in blue and in red so there's things in red that we could potentially add through the pipeline, but we don't have a way of doing that yet. You can kind of get an idea of what we're passing to the pipeline, and then you'll be able to see at the end what happened, what got filled in, what didn't. So just have a quick scan about what's happening within the pipeline. You can see there's lots of different processes that are happening here. You can see how many of them have completed. Lets you have an idea about what's going on. Let's go back to the presentation, and I can kind of explain a little bit about what the pipeline's doing. So I restart the slideshow. So I showed you the sample sheet. Essentially, yeah, it's just information about the samples that we're using within the analyses. So you can have multiple samples of different types in here. They just have to be arranged in this format with the 3 columns with the sample name, the type of data, and the file location. This is what I was saying about the template. So I showed you the Word doc, the docx, the Word version of the template, which is what we're currently submitting to F1000, but we're also working with F1000 and Wellcome Open Research to produce an XML version of this template as well. So there's different options for how you provide the template. So the way that Nextflow works is you can break down sections of work into sub-workflows, and that's kind of what we're doing here, which means if you've got repeated processes that are shared across different pipelines, you can put that into a sub-workflow, and then you can share that sub-workflow across different pipelines. So for example, things like running Busco, and which is something that we do in several of our pipelines. We could have a sub-workflow that manages that, and we can share that code across the different pipelines. But so I'm just going to go through the individual sub-workflows that we are using within this, and it kind of give you an idea of what they're doing. So the first thing we do when we run the pipeline is that we check all the inputs. So there's two kind of main inputs here. So there's the parameters that are provided and the sample sheet. So for the sample sheet, we'll go through, we'll check that we can find the files that were listed and that everything's okay with those. For the parameters, we will check that it's a valid assembly accession. We'll check that we can find it within ENA. Same with the biosample, we'll check that the biosample accession exists and that it corresponds to the same taxon ID as the assembly to make sure that we're not mixing data up. And we'll do the same thing for the biosamples. And we'll also use these to fill in a little, to fetch a couple of other bits of information like the taxon ID for the assembly, the bioproject for the assembly. These are just things that we then need to be able to run the next steps in the pipeline. So yeah, so this will just run two processes. This is kind of the way that Nextflow represents how their pipelines work. The individual modules are marked in these kind of ovals. And where you see a module in white, it's a module that we've written within the Tree of Life. Anything in green would be a standard NF core module that someone else has written and we're just reusing. And anything in this, it's not showing brightly here, but that's a kind of a grey module, is a module that we've written, but it doesn't always run. It's an optional module, and that will be controlled on the kind of input that you've given to the pipeline. So whether you've requested for a certain type of analyses to be run or not. So one of the main things I've worked on with the pipeline is fetching the genome metadata. So with this, we have a list of files that are contained within the pipeline in the assets directory, which are links to APIs at ENA, at GOAT, at COPO, which let us kind of take a taxon ID or an assembly accession or a biosample accession and go away and query those resources to ask for information about those things. And from that, we'll get a file back from them, which we can then process and pull out all the information that we want for the Genome-Note. So we use the input metadata, we combine that with the list of the files, and then we run a series of wget commands, which is basically just a way of making an API call. So we'll go away, the pipeline will say, get me this file. We'll do that for all of the files that are mentioned within this template. We will then parse those, we'll open those files, read them, pull out the information that we want from them, and write them out to a file. So each of the different resources will produce its own file, which has a list of parameter names, which are essentially the placeholders that you saw within the template, and the value from that source. We do a slightly different thing when we're fetching data from GBIF because we have to make several API calls rather than one to get all the information that we need from GBIF. So that has its own module to handle, but the rest of all the metadata sources we handle with these processes. And then the final step that this metadata sub-workflow does is it takes all of the output files from these processes and combines them. So we go through all of the data from the different sources. We find where, so for example, if ENA has a biosample accession, and NCBI has a biosample accession, and COPA has a biosample accession, or a value for who collected the sample, we'll kind of compare all of the information that's the same kind of type from the different sources. And if all of the sources agree, if the value from each of the sources matches, we'll put that into a file for consistent data. But if one of the sources has a slightly different value to the others, that will go into a separate file for inconsistent information. We still want to keep that information. We want to report that back so that the editor can check it afterwards and see what they will want to include in the Genome-Note, or if there's something kind of gone wrong with the metadata submission from one of the sources, and we can get that fixed. So yeah, we get sent essentially a set of metadata parameters, and we know where they've come from and what the values are. Then we also have the genome statistics sub-workflow, and this does more of the computing of the statistics and the assembly metrics. So we have input to this. We give it the assembly, the genome FASTA file. We give it the unaligned PacBio reads, and we can give it information about lineages to allow it to run Busco. I probably should have said this, any of the parameters, kind of anything here in black is something that gets supplied by us. In blue, it's an optional input. The orange is also an optional input, but there's a default value. So you can override this, but there's a default, there's something in the pipeline that it will use on its own to kind of do this. So within the genome statistics of workflow, we heavily use the NCBI data sets. So we'll use that. We actually call summary genome twice, once to get metadata on the assembly, and once to get the kind of the chromosome list, which we use in several places in the pipeline. NCBI get ODB, I think, is used to get the lineage information that we use for running Busco. And then we run Busco, which is gives us an idea of the kind of completeness of the assembly, and gives us scores for kind of gene counts. That is one of the steps that takes the longest time within the pipeline. We then do once Busco is completed, we kind of tidy up the output from that and kind of formulate that in a way that we want for our output. We run FastK and Mercury to generate completeness scores, QV scores. And then the final step is pulling all the information from these different things into a table, which is one of the outputs from the pipeline. So that's basically the statistics that we calculate all go into that table. We have the contact maps sub-workflow, which we use to produce the Hi-C map. So that takes the chromosome list that we've generated earlier in the genome statistics. That's a workflow, or if there was one supplied as a parameter, that will come in here. This uses the aligned Hi-C reads from the sample sheet and the genome FASTA from the minus FASTA input. And that runs a series of tools. Quite a lot of these are standard NF core modules that we've been able to reuse. And actually at the bottom here are two optional steps, which were kind of designed to make our life easier within the tree of life. So the output from the contact maps workflow is 3 files, which can then be uploaded to a HiGlass server to produce a Hi-C map. We've written 2 kind of helper modules that will automate that process for us, but they're kind of only really designed for use within our project. If you have your own Kubernetes hosted HiGlass server, then you'll be able to use them. But yeah, the idea is that they're kind of there to make our life easier. And then kind of the final step in the pipeline is pulling all of the information together from the different sub-workflows. So these are the files that I was talking about earlier, the consistent parameter file, the inconsistent parameter file. We also, there's the parse metadata, the first step here, that step got run for all of the information, all of the files that were produced by the genome metadata sub-workflow, but we also run it now again on the output of the genome statistics sub-workflow, and that gives us the statistics, the parameter, the values, the placeholder values from the statistics sub-workflow, and we have this, so we end up with 4 different files. We have 2 files where we have consistent parameters, and then we have 2 where they are not, where they differ, and we'll run some steps that will kind of pull that information, and we'll end up saying, okay, this information that came from the statistics of workflow is the same as the information that came from the metadata sub workflow, and we'll use that to run the populate template step. And that's what generates the final document for you. And we also have an optional module that will write the parameters to a database that is, again, that's internal tree of life use. I'll talk more about that at the end of the session. So in terms of pipeline outputs, when you run the pipeline, you'll notice that there'll be a set of files that get produced in your working directory. There'll be a directory called work, and this contains all of the files that are used by Nextflow as it runs the pipeline. So all the logs of the different steps will be there. You can kind of have a look and see what commands will run. You can find out if there are any errors with the step. The outdir will be where the results end up. So you can specify that, so you should be able to say that. And the Nextflow log is just general logging information. So the results in the output directory are going to be described in the next slides. So we talked about the contact maps. So within the results there, you'll have contact maps with 3 files. And if you upload these files to a HighGlass server, you'll produce a Hi-C map. And we do this for all of the assemblies that we produce within Tree of Life. They go to this server that we maintain here, and we add a link to that map within each of the genome notes. Within the genome notes document itself, we'll go and have a look at the results from the pipeline in a second, but you can see the docx, which should be prefixed with the assembly accession, is your kind of completed genome note document from the pipeline. And you'll also have the consistent file and the inconsistent file, so that you can have a look at the variables that we used to fill in the pipeline, to fill in the placeholders, and any that weren't used because there were slight differences between the different sources. And often within the inconsistent file, it's minor things that are making them different. Sometimes one source might, if it's including something like collection information, it might include an additional bit of information that the others don't, so it's not quite, doesn't quite match up. Or sometimes I think we see with scaffold counts, there may be slight differences in the way things were rounded up. Yeah, so the idea is the docx output that you get from the pipeline, you can look through, you can see what's been changed by the pipeline. If there's something you don't agree with, or if the wording wasn't quite the way that you would want it to be, you can go and edit that. You can make any changes that you want to that document. And there's also the assembly.csv file, which is all of the statistics that were generated with the different steps. Busco, that just contains all of the Busco information from running the pipeline. I'm not going to go through all of these. Multi-QC, so that's kind of something that comes as standard with NF Core, I believe. It's just a visualization tool that kind of generates a report summarizing all the samples in your project. It's not something I particularly use. It's not particularly It's not relevant to the production of the genome notes, but it's just something you'll see within the output directory. And then you have the pipeline info directory, which just kind of gives you a report about what happened, how things ran, what resources they took, how long different steps were, and you can kind of track this information. And we do this, we in production, we kind of keep a record of pipelines that we've run so that we can look at things like making sure that we're requesting the correct resources for running the pipeline so that we can be kind of more economical with energy usage with the requests, make sure that we're not asking for too many resources on the cluster. It's just kind of trying to make it as streamlined and as efficient as possible. So I'm just going to exit the slideshow and go back to the Git pod, and we can have a look at the output. It should have finished running by now. Oh, maybe it hasn't. Okay.
Yeah, the last run that I did took 35 minutes. So I'm not sure which time we started that.
Okay.
Around 2:30, so it'll probably need another 10 minutes.
Okay, that's fine. That's a little longer than it has been doing. Go back to the slideshow. Okay. So I mentioned earlier that there are other pipelines that we use to generate content for the Genome-Note. So there is the Sangatol Blob Toolkit pipeline, which we use to generate Blob Toolkit plots, and these get included within the Genome-Note. And they also get uploaded to a Blob Toolkit viewer. So you can kind of interact with them here, and yeah. And the Sangatol read mapping pipeline. So I mentioned that for the whole genome sequencing, sorry, not for the high C reads, the pipeline expects the reads to have been aligned to the genome. So we run the Sangatol read mapping pipeline for each of our assemblies before we run the Genome Notes pipeline, and use the output from that as an input to the Genome Notes pipeline. So this is kind of how you can link different pipelines together to go from raw data, raw submitted data to a Genome Notes publication. So NF-Core have a pipeline called Fetch NGS, which you can give it a set of read IDs, a set of read IDs, and it will go and fetch that information for you from ENA or NCBI, and download the information. When I've run the pipeline on assemblies that are not produced within the tree of life, this is how I would go about it. I would run fetch NGS to get me the sequence and read information. I would run Sangatol INSDC download, which fetches the assembly FASTA file. I would then run Sangatol read mapping, giving it some of the output from fetch NGS and from INSDC download. And then that would provide me the high C data that I need for running the Genome-Note. I would use the Blob Toolkit pipeline would be run to generate the Blob Toolkit plots, and that uses information from Fetch NGS as well. And then as actual inputs to the Genome Notes, would use the reads that are produced from Fetch NGS, would use the aligned reads from the read mapping pipeline, and we would use the genome FASTA file from INSDC download. I've also added in the Sangatol ensemble gene download. That's not something that we're using yet, but it's something that we're looking to do when one of the extensions that we want to make in the near future to the Sangatol Genome-Note pipeline is adding in information about the annotations. So we're working on a new sub-workflow for that, which will take in the GTF files produced by Ensembl and allow us to work out some kind of annotation statistics and metrics. And we can then include that information within the Genome-Note itself. So that's kind of, and we're kind of designing and developing these pipelines so that they work together, so that the outputs from one is suitable for the input to the next, so that we can chain these essentially. So yeah, I just mentioned we got some version 2.0 of the pipeline was only released this week, so this is quite new what we're showing you with the templating and the Genome-Note document being produced. But there's lots of things that we're currently planning to do. So one of them is the annotation statistics of workflows. So that's looking at any kind of annotation provided, whether that's initially we're looking at Ensembl data, but we can also extend that to look at annotation provided by RefSeq in the future. We're looking at adding additional or alternative tools for assessing the genome assembly quality. So this is things like GFA stats, Flagger, Genome-Scope. There's a lot of these suggestions have come from other groups that I'm talking to that want to produce their own genome notes. So if there's anything that you think should be included within the Genome-Note, please let us know, and we can look at adding it to the pipeline. We're also looking at adding calculations for genome and transcript mapability to the pipeline. So from our pipeline's website, you can find the GitHub link, and you can see the kind of issues that are reported. Actually, I can show you that. I go to here, there's a link to the GitHub. And then we kind of track issues. So if there's a feature that you would like or something that you think's wrong with the pipeline, you can create a new issue and talk to us there about it, and we can look at adding it to the pipeline. So in terms of the other aspect that I've not really mentioned too much about is, as well as the Nextflow pipeline, we're concurrently working on developing a platform for producing a Genome-Note. So the idea is that there will be a web interface, and anyone can use this to then go away and generate their own Genome-Note content. So you would log in, you would request a new Genome-Note, you would give it some basic metadata. Sorry, the screenshot's a bit blurry, about the assembly that you want to kind of produce the note for. So you start with a taxon ID. From there, you would select the assembly that corresponds to that taxon ID. You'd specify the samples to use for the analyses and the reads to use for the analyses. And then the web interface would trigger running the Nextflow pipeline. The pipeline would run, and all the information from the pipeline would be written back to the database that sits behind this this web platform. And once the pipelines have run, you'd kind of be notified, and you would be able to log into the website and see the Genome-Note. And there would also be the option, you'd be able to edit the Genome-Note within the platform, so you'd be able to add author information, references, and essentially make any changes that you want to then output the final document. So yeah, that's kind of what we're working on. That the idea is that anyone will use it. You can. Oh, the other thing you can do with the platform is if you want to use a different template to what we have, you'll be able to supply your own template through the platform as well. So you can produce a Genome-Note of your own format, and have it output the document that you kind of require. There would be ways for you to share the document with other people. So if you've got several people that contributed to the Genome-Note and you want them all to have a look at it before it gets submitted, there would be ways to kind of preview the document for others to kind of see it. And we want to work with F1000 to allow automated submission from the platform directly to them. So we would completely bypass the submission forms and kind of save time there. Okay. So one of the other things that we do within the Tree of Life is we host a site called the Genome After Party. It's a public repository of the core genome analyses which result from running all of the Sanger toll pipelines. That's hosted at Sanger on our public S3 server. And this isn't just DTOL genomes. This is all of INSDC with new data being added every week. And you can see that all of the information, this is just an example species here, all the information from running our pipelines, including the Genome-Note and the genome statistics, are added to the after party. So you can find all the information relating to a genome from the after party site. Okay. Any questions for me on the chat that haven't been dealt with?
There's one in the Zoom chat asking whether or not the Universal Genome Notes platform requires any coding experience.
The platform itself, no, shouldn't do. It should be the only interaction. So the pipeline, the platform would essentially be a UI. You would be able to kind of make any edits there. But yeah, it would run the pipelines itself. You wouldn't need to know anything about those. You should just be able to supply it with some basic genome metadata, have it run the pipelines, and it will produce a document for you, which you can kind of view on the platform and edit. Edit selected fields. We wouldn't allow things like the assembly statistics to be edited because obviously we don't want people changing those. But yeah, it wouldn't require any kind of programming knowledge at all. I think the thing that I didn't say here is that the pipelines would be run on the Sanger infrastructure, and there would be the option to have the data uploaded to HighGlass. So yeah, someone's asking about the Universal Genome Notes platform. It's still in development. It's not available yet. It's still actively being worked on.
So does that mean your data already needs to be up on a public database prior to running it?
It does. That's the only restriction. So to running through the Universal platform is we require all the data to have been submitted. If you want to run the pipelines on data that's not public, there is a way of doing that. You can override you can specify some of the parameters rather than have it fetch them. And as long as you've got your files locally, you can give paths to the files and you can run the pipelines that way. But the platform itself is being developed to work with public data. Okay. Okay. So the pipelines
quickly wait for Busco.
Yeah, there's things I can show you that because some of the steps have completed, so I can show you some of the intermediate outputs that have happened. So I look for the metadata. what I'm doing is I'm looking at the Nextflow log file and I'm finding the process that I want to see, and it will tell me here what the work directory was. So I can then look within the work directory and see what the outputs and the inputs to that process were. So I can kind of show you. This is from, this is all the metadata files that were produced from querying the different data sources. So you can see that we queried COPO with a biosample for the high C, an ID with it for the, sorry, we queried COPO with the high C biosample, we queried COPO with the RNA biosample, and we queried ENA for assembly data, we queried ENA for taxonomy data, and then the outputs, all of these files kind of, oh, I keep forgetting that you have to specify to open it with the test data. Is the output from querying COPO with the high C biosample. So what we get in each of these files is we get a parameter name and a value. So this parameter name is the placeholder within the template file, and the value is what COPO has for that. So for example, you can see high C, the common name for the sample, high C, COPO has that down as broom moth. We've got collector information, we've got a collection method, we've got a collection location, and all this information would then be taken and put into the template. But because we've got lots of different sources, which all kind of will have similar data, we want to compare them and make sure that it's consistent. So what we end up with, once that comparison step is complete, we have a file which again gives us the parameter name and the value, and it also gives us information about where that information came from. So you can see here for the assembly session, all three ENA and NCBI had the same value. So you can kind of see this isn't information that makes its way through to the template, but it's information that you, as a pipeline runner, can kind of go away and check. So this lets you see where the data's come from, what the values are, and kind of how much support it has across different sources. And what's actually possible. So this is all information that will then go away and be put into the template. But we also have an inconsistent file. Let's have a look what's in there. So here you can see that the collection location for the biosample is not the same in different places. I'm not sure what the difference is. Okay, so we can spot here that COPO have got United Kingdom as part of the location, but Scotland and ENA don't include that. So it's not matching, it's not an exact match. So that's something that's fairly easy to reconcile. It would be up to the editor then, in that case, to take that parameter and add it to the document with or without the country name, depending on what they want. We have a difference in contig numbers. I think that sometimes happens if the mitochondrion is included or not. So I think that's probably what that is. This would normally, we normally kind of correct this in the next step where we merge the metadata with the genome statistics, and we would kind of take the value from the genome statistics. So I think you'll probably see that number does get filled in the template, but there's an additional comparison between this file, which is the output of the metadata sub workflow, and the output of the statistics of workflow, and they kind of get combined and added to the template. So there's some actual rationalizing steps that we do. So with this country name, we've got some code within the process that does the populating of the template, that does a little bit of checking prior to populating the template, and that would be kind of fixed there. What we would do in that case is we would return the longest of the strings, because it's more or less the same, just with the country, the UK bit missing, and then we would let the editor kind of make any changes that they want to the document. Let's see what else we've got different. Yeah, again, I think genome length is going to be because one includes the mitochondria and one doesn't. So from there, this is kind of, you can see the outputs we have here. Within the results directory, this should be, where do we put the results? Let's write that down. BGA test results. You can see that the contact maps files have been produced because those steps have finished. We've got some kind of pipeline info that just tells you what's running, gives you some statistics on what happened. The sample sheet valid is, that's output from the input checks. I think that just shows you the CSV file, yeah. So what we can do while the pipeline is running is we can have a look at the actual genome note that was produced for this test species. So we kind of open that up. There is a link to it within the Gitpod page, within the simple browser, if you've got that open. If not, I can link to it, put a link in the chat. Discord. So you can see we've got a specimen image provided. We've got all of the information. So this template's going to look slightly different to what I had in the slides earlier. Those slides are a little bit out of date when we were going through the Genome-Note anatomy. There's quite a few improvements that have been made by our Genome-Note editor to how we format our genome notes since then, including the information that we have about the method sections. So, yeah. It was probably more helpful. There's a little bit more information about the template. If I go back to Genome-Note and open up the template, I can kind of show you some of the things that we do. And then once the note's been produced, you can kind of have a look. So within the template, why not scroll? You can have optional bits of text that only get included if a parameter's valid, for example. So you can see here we've got in red this end if. So if annotation URL exists, we would include this sentence in the template. So this is basically saying if we've got an annotation URL, gene annotation of this assembly on ensemble identified this many protein coding genes. We're not currently pulling in annotation information within the Genome Notes pipeline, so you won't see this in the output Genome Note, even though there is assembly annotation for this species. This is one of the things that we're looking to improve within the next release when we add the annotation statistics of workflow. Let's see what else there is. So we have optional sections. So for example, if there is a high C specimen that's different to the whole genome sequence specimen, we would include the section. Same with the RNA-seq. So there's this merging the template parameters in will kind of handle a lot of this for you. You can build quite a lot into the template to kind of control what happens and what you get out. Are there any kind of questions while we're waiting for the pipeline to run? Any information from any of my team that I missed that they think I should cover?
There's a question in the Zoom chat asking how long it typically takes to run the pipeline.
This is only a test sample, so this one normally runs in about 10 to 15 minutes. So I'm not quite sure what's happening today. It very much varies though when you're running for the entire assembly, for the full data set. Sometimes it's a few hours, sometimes it's a day. I don't think we've had many that take much longer than that. I've not been doing the production runs, so I don't have that data, but I think most of them are within a few hours. So like maybe 6 hours, 7 hours. It's very dependent on the genome size.
Yes, I don't think I've got average statistics right now, but I think the average is far below 12 hours. In our production system, we got an alert. If pipeline takes more than 24 hours, we get a message. I don't think I ever saw such a message for Genome-Note. So yeah, it's relatively quick.
Yeah.
I think Bet you're unlucky because my run on Gitpod finished
Just finished.
10 minutes ago.
I can say I've got the document. I can share it. It just won't be what's actually from this pipeline run. I've got it saved on my computer from earlier. Is any of the anyone that's running it have your pipelines finished? Are you able to have a look now? Okay. I can open up a previous one. Making sure I open the right one. Yeah, okay. So can you kind of see what we have here? Make that a bit bigger. When your pipeline runs, you should see a document that looks like this. It would be within the BGA test results. There would be a folder called Genome-Notes, and there would be a file that looks like this in there. And you can see the author list has been filled in. The way that we do the author list within the Tree of Life Genome-Notes is the reason that we're able to semi-fill this in is we always include the sample collector first. So in this case, there were more than one sample, so we've got a list of the different collectors, and we try and do some clever formatting on that, but we need to do that. Some improvements to that, so you can kind of see that authors now has values in it. Anything that has been filled in stays in green, so you can see what's changed. You see we've got the common name, we've got the taxonomy information. We don't have sample number and sample sex. That's probably something we should be able to fill in. We've got the locations where the samples were collected. Observed sex isn't something that we're fetching with the pipeline at the moment. We're filling that in from our internal databases. So that's something that our editor would be able to add along with all of the recounts. This is something that we think we should be able to pull in through the pipeline, but we're not doing yet. So it's another potential improvement to make. You can see some of the assembly statistics have been filled in. So the assembly length, the sequence number, the scaffold number, the number of gaps, the scaffold M50. the QV score, because we're only dealing with a very small amount of data. Let's have a look here and see how this compares to what was actually published. Trying to find the same section. The QV score that we're getting is quite different. There we go, that's what I'm looking for. So here we have a QV score in the report in the published Genome-Note of 68.5, and became a completeness of 100%. We were only using a subset of the data, so we don't have a valid number here because we only had 2 chromosomes and a very small percentage of the reads that we were using in this test. So that value is not valid. Same with the BUSCO. That's not a valid data. This is just for test purposes to get the pipeline to run. But if you were to do it on a full assembly, we would get a BUSCO score that would look the same as here. So yeah, you can kind of see that the pipeline runs, it fills in quite a lot of the information. There's still work to be done, but it goes quite a long way to producing a completed Genome-Note. And the system itself is quite flexible. The template that you provide will show you a Word document here. We also can work with XML. You can kind of make changes to the content of the Genome-Note if you want to. So for example, one of the groups we're working with want to include a kind of discussion section, so they can kind of add that within their template. That's much harder to automate though, because that's going to be very dependent on the assembly, but there's perhaps things that can be done. They can include different assembly metrics. If you had the same set of authors for every Genome-Note, because our author list varies for each assembly, you could kind of have that standardized within your template as well. So that would, and the same reference list. Yeah. Oh, I'm on the populate template step now, so pipeline is almost run. And the final thing the pipeline does is it dumps a table of all the software tools that we used within the pipeline and the versions that we use, so that you can kind of share that information and have the pipeline fully, results fully reproducible. Running multi-QC. If I have a look in my, then the pipeline is finally finished. And that took a lot longer than I was expecting. Sorry about that, everyone. So within the Genome-Note directory, you can kind of see here, you've got the, oh, that's the wrong way of doing it. You can see all the parameters that were filled in. You can see the ones that weren't used. In fact, in this case, everything kind of was added in. You can see the outputs from the genome statistics of workflow. So this is where you get your chromosome list from. We do some nice formatting on that before we add it to the template. So we kind of do some rounding on these numbers. There's your BUSCO score, QV score, the completeness, and then the completed Genome-Note document. That's kind of should hopefully match up to what I'm showing you here. But yeah, so that's, you can see whether yours is finished or not, it should work eventually. Hopefully that was useful and gives you an idea of what we're doing, what we're working on. So question in the chat, so what are the arguments for sidestepping peer review on the Genome-Notes? So there's kind of 2 ways we're looking at this. We're looking at the automated kind of metrics can provide one review. And we're also talking to Wellcome Open Research about a different way of doing peer review for genome notes, because they're not your regular kind of publication. They're very much standardized. They follow the same format. Quite often when we get reviews back, the comments are that there's not a lot of kind of comparison between other species and not a lot of additional information. So we're looking at possibly setting up review panels that would be able to kind of mass process a set of genome notes. So long as the genome notes they serve kind of a different purpose. They're there to kind of announce that the assembly exists, to acknowledge everyone that's involved, to give credit, to kind of say that the assembly is of a good enough standard, and to let people know where the data is. So we're looking at maybe kind of changing from the traditional peer review to having the automated peer review, and to having them assessed by a panel at once that understand what a genome note is. Ritu, do you want to chime in here? You might be able to explain it better.
I don't know if I can explain it better. I'm not even sure what else to add to what you said. Yes, it's maybe we could say that the platform itself and the pipeline are sort of journal agnostic. So each journal will have its own format for notes and its own review process. So wherever you submit journal, you have to adjust your expectations there. And this concept of review panel, that's something we're investigating with F1000. Other journals may want to do differently. And similarly, the extent of the review, so whether like sidestepping and whether they understand the ascending metrics are a proof on their own of the quality of the assembly. That again is an agreement with the editors and journal.
Okay, hopefully that answers your question, Katya. Are there any other questions in the chat?
I think it says Katya's still typing, so there may be a follow-up.
Yeah.
Oh, okay, cool.
Okay. Well, I guess that's everything I had. If there's nothing else anyone wants to discuss, I'm happy to end the session there and give people some time back. Or
One has just popped up in the Zoom chat.
oh, I've lost the Zoom chat.
For automatic review, could you share what type of metrics you think would be useful?
Oh, the automatic review. So they are the metrics that are in the BGP paper, which I had linked in the slides. So I think it's things like, let me go back. Let's see if I can find the link and see exactly what they are. It's things like the QV score. Is Busco part of it?
Yes, there's the assessment of the completeness of the assembly. Verbose go something else.
It's not something I know off the top of my head, but it's certainly something I can add. Here we go. Config N50, scaffold N50, QV.
And percentage assigned chromosomes.
Yeah, so yeah. So these are all things that kind of appear in table 1 on our Genome-Note reports.
It's now table 2.
Not table 2, sorry, it's changed, hasn't it? There has been talk about, so yes, it's these, the assembly metrics are here and the benchmarks are part of it as well. So we have the KV score, the KMA completeness, the BUSCO score, the percentage of the assembly that's mapped to chromosomes, and then any information on sex chromosomes or organelles. We have been talking about splitting the assembly, the quality metrics into their own table to make it clearer that they are kind of what's being compared for the manual review, but that's possibly a template change that will be happening within the future.
They've clarified their question. They meant for the automatic peer review panel.
Oh, for the peer review panel. I think it would just generally be a case of making sure that, that the assembly reaches the quality metrics that we've defined here, that the data is available, that it's crediting everyone. I'm not sure what else gets reviewed, to be honest. Yeah, I think it's essentially just that the Genome-Note is fit for purpose. It's reporting all the information that we kind of stated that we should be within here. So yeah, it's announcing the availability, it's assessing the it's giving an assessment of the assembly quality, and it's acknowledging everyone involved. I think that's kind of the purpose of a Genome-Note, and that's really what we want the review to check that. Yeah, that's something that could potentially be possible. I think that's something else that we're investigating is publishing in a slightly different format to the traditional Wellcome Open research, where we would have we're in talks with PubPub. I think perhaps Mark and Harris will talk more about this as part of the BGE later this month. But one of the things we're talking about with them is if we were to kind of investigate publishing genome notes there, we could have kind of like a comment forum to allow people to discuss the assemblies and kind of give information that way. But yeah, this is still something that's being worked on because the scale of the assemblies that's going to come over the next few years is intimidating. So yeah. But yeah, I think there's better ways of doing it than currently are in use, just in terms of scaling. But yeah, something like an open kind of forum for review and would be definitely worth investigating.
There's been someone has asked for a quick brief on what the genome after party is. They may have missed it earlier on.
Okay, so the genome after party is basically a tree of kind of the outputs from running all of the pipelines within the tree of life. So that's not the one I wanted. Basically, we run our pipelines on all of our assemblies, but not just all of our assemblies, any of the INSTC assemblies. So it's basically a repository of the outputs of running all the pipelines. So things like making the outputs available for free to use for anyone to so that you can search for a genome, you can see the kind of data that we have. What's the dolphin, wasn't it? So yeah, you can have a look. You can, rather than individuals having to go away and rerun their own pipelines to compute what will essentially be the same data, it's a repository. You can go away, you can pull the aligned reads, you can get the statistics, you can get variant calling information, and we do this and make it available for all our assemblies and for anything in INSDC, I think is the aim eventually, start pulling things in. But yeah, it's essentially just stop people having to do the same analyses over and over again, just to have it done once and shared. Oh, there's the chat.
Okay, cool. Shall we have last call for questions now then?
Sure.
And then we can finish up. Feel free to turn on your mic and just shout. Don't have to be scared. Right, in that case, thank you all for coming. Thank the three of you for giving up your time and giving the session. And hopefully we'll see you all in the next one. Goodbye.
Bye.